We're continuing to work on chapter 15. We have learned about fatty acids and then we learned about triglycerides. Now what we're going to look at is phospholipids, which are a type of lipid that contains an ester. So we'll be looking for those esters right off the bat in our phospholipids. So as I look at the slide with pictures of phospholipids here, when I see this phospho, that prefix tells me that it contains phosphorus. And we're going to see that these molecules look a lot like our triglycerides with a slight modification. So I'm going to draw the same box diagram we did for our fatty acids. We have, again, a glycerol backbone, and we have a couple of fatty acids. So you can see these long carbon tails. This one has a bend in it, so I'm going to represent that when I draw it as well. It's not just a nice straight fatty acid. And then here's the difference. In place of that third fatty acid, now I have the phospho piece of it. So there's phosphorus, there's actually a nitrogen in there too. And in that head, if I can help you see it, there's a positive charge and a negative charge. And so this phosphate or phosphorus head has both positive and negative charges on it. And that means, if you can think about it, what does that mean for water solubility? Those charges, we can say charges on the head make it very water soluble. So these compounds are going to be very water soluble, make phospholipids very water soluble. Charges have enormous impacts on water solubility, and so we're going to keep an eye on those as we look at these structures. So that's our kind of box diagram. Another way that you might see these phospholipids drawn would be kind of like you see here, where we have a big polar head, but now we'll have two tails, and sometimes we'll see a bend in those. So this would be a boxes, this shows all the atoms, and this would be another even like kind of simpler cartoon picture. We have a polar head, and we have these non-polar tails. And we said a couple minutes ago that there are esters in here. So let's look for carbonyls and see what we can find. Here's a carbonyl with an oxygen, and here's a carbonyl with an oxygen. So these, again, have esters in them, and they also have this double bond here. So these we're not so interested in breaking. The main place we're going to look at using these phospholipids is in a lipid bilayer, so in a cell membrane. This is a picture here of what we call a lipid bilayer. And these polar heads would be on the surfaces. So all these little yellow balls, right? Those are these polar heads here. And that would be this whole region or this whole region. Those are going to be on the surface of my cell membrane. So on the outside here. And here's kind of a cross section of this cell membrane as well. Anywhere it's yellow, those are my polar heads. In the lipid bilayer, if you can think about this, there's water on the outside of the cell, and there's also water on the inside of the cell. And so these charged heads are able to form attractions with water and able to form attractions with water outside of the cell and inside of the cell. And then in the middle here, these tails are all these carbon-hydrogen tails. And so all of these tails, both here and here, are going to be very non-polar. So I would say that the interior of this lipid bilayer is non-polar. That's an important thing to look at. If I have these polar heads, or these positive and negative heads, I have ion dipole attractions holding the water and those charged heads together, right? So the attractions are ion dipole attractions that I'm looking at right here. Inside here, where I have all those nonpolar tails, here I have dispersion forces, right? And so that would just be these temporary weak attractions between all these nonpolar C's and H's that are floating around. And together, that attraction to water and then those interior attractions to each other are holding this lipid bilayer together. A couple more just little features I want to show you. This bend here, you might have heard of this lipid bilayer in animal cells at least, called a fluid mosaic. And the fluidity of it is different from very rigid cell walls. And so if I can think about my saturated tails We had learned back in chapter 11 that these saturated tails tend to glob together and form solids, and we don't want solids as our lipid bilayer because we want gases to be able to just diffuse through, for example. We don't want to break our cell walls every time we push on them or touch them. So the fluidity actually comes from this bond right here. Can you tell, is that a cis or a trans double bond? So you see the H's together on the same side. So if I put this cis double bond in here, that cis double bond shows up in a lot of the tails of our phospholipids, and that's why we have this little bend in it, because that bend is coming from a cis double bond. This makes my cell membrane more fluid. So it makes the cell membrane more fluid because it reduces some of those intermolecular forces, right? It weakens those attractions. Because it's more fluid, it's also it's also more flexible, and it's also more permeable. Permeable being a word to say things can permeate through or things can get through it. So that cis double bond that I see in that bend keeps these interior tails from sticking together too much. As I look at this picture here, we just want to pick out a few different features of it. So I have all these phospholipids that I colored in yellow. There's a few oddball molecules in here, and let's see if I can help you see them. They're very hard to see when I have just the black and white here, but they don't have any tails on them. And so there's some random blue ones scattered throughout here that don't have tails. Those are cholesterol molecules. And cholesterol is good for adding a little bit of rigidity. So we don't want this to be totally oily to where it just falls apart. So a little bit of having, we'll see on the next page what the structure is, but a little bit of cholesterol adds some rigidity. Other things that you can look for in here, glycoproteins are gonna be sugars with proteins. Over here, a glycolipid. Glyco is going to be a sugar with a lipid. So this is the carbohydrates, the branches like we saw in chapter 14, and then they're connected to one of these phospholipids. In a glycoprotein, you have a protein instead, and then some sugars connected to it. So we'll look just a tiny bit more at these cell membranes as we continue this semester. As I look at the different ways of getting across this membrane, if you're small, you're able to just move by simple diffusion. So that would be, for example, oxygen or carbon dioxide could just move with simple diffusion. If you need a channel to get through all those nonpolar tails, then you might have facilitated transport. And you can see here that this is a protein, kind of like a protein tunnel for things to move through. And then say you're trying to pump a solute to where it doesn't want to go, like maybe move potassium and sodium in directions that they wouldn't normally diffuse, then you might have that protein channel and you might require ATP or some energy, and we would call that active transport. So 3 different ways of getting across this cell membrane. If you're too different from these tails, you need probably some kind of tunnel to help you get through, and that tunnel is going to be a membrane protein. And you can see those same protein channels over here as well. So that's as far as we're going to go with phospholipids, be able to recognize these different features and know why that cis double bond is there as well.